All right, I don't have your exams ready. Obviously, I kind of read 170 exams. Taking me time, and I'll get them back to you as soon as I can. No promises. All right, so we talked about B cell development, and we're going to continue today. Now, this is a topic which is in a state of flux. Our understanding what's going on as far as how B cells eventually can produce an antibody with the help of T cells is still lots of question marks. A lot of what we now understand to be going on in the lymph node is because of microscopy, which can take movies of cells moving within the lymph node at different times after antigen was introduced into an organism, a mouse, for example. And what you do is you fluorescently label the different types of cells with different colors, and you can follow them in real time where they're actually going. So it's actually quite a fascinating technique. And your book actually has some places you can go to see these movies. Well, the movies are interesting, but they don't really tell you what's going on unless you know how to interpret what the dynamic of those movies actually is. So we're just going to look at these plain cartoons instead. But another problem with not understanding what B cells do in the lymph node along with T cell health and APC is that at different times B cells move back and forth in different locations within the lymph node. And that's because people have discovered upregulation and then downregulation of chemokine receptors, and then in addition to that, chemokines being produced by different cells cause the B cells to move back and forth within the lymph node at different times, which begs the question: how are these receptors being upregulated? How are they being downregulated? And how are they being upregulated yet again? So it's a fascinating area of research, which is ongoing, and a lot we don't still understand about the timing of these receptors and the timing of the chemokines, and again, what brings about the movement of the B cell back and forth within the lymph node. So some things are going to seem somewhat, I hope not, but may be confusing again because the B cell is going to be, again, moving in different places and then returning to the place where it just came from. So it's going to seem redundant, but I'm hopeful I can clear it up as well as I can in this lecture. Just as a review, this is the gross anatomy, if you will, of a lymph node. We have two main structures that we're interested in. I'm less interested in the medulla, but we have the cortex, which contains the follicles, where B cells will eventually become able to give rise to plasma cells, which, as you know, will produce the antibody. And that's occurring in these follicles. And a primary follicle is these little things. Nothing much is going on in them. They do have the architecture, however, to allow B cells eventually to become activated, and I'll tell you what that architecture is. But in any case, not much proliferation is going on in the primary follicle. However, once a B cell becomes activated, it starts proliferating, having progeny, and that becomes a secondary follicle. You can see it's larger. And in the center of the secondary follicle is the core area called the germinal center, which we'll have more to say about. Interior to the cortex is the paracortex, which traditionally is called the T cell area. But I hate that description. And the reason I hate that description is because there are B cells in the paracortex, and there are antigen-presenting cells for sure. So to call it exclusively a T cell domain is misleading, immunologically speaking. So we're going to, again, look at what happens at the paracortex as well as at the cortex today. And then finally, the medulla. Medulla is where cells enter this part of the lymph node to actually leave. That's where the efferent lymphatic is. So cells can actually leave. Antibody products can also leave, obviously soluble in the lymphatic fluid. They can leave the efferent lymphatic. And plasma cells can either reside and stay in the medulla, or more commonly, really, and this gives you the wrong impression, that plasma cells only stay in the medulla to secrete their antibodies. But more commonly, plasma cells leave the medulla and make their way to bone marrow, where they take up residence, and that's where they can either live a short period of time, throwing out antibody, or actually some plasma cells can live for decades and produce antibody for many, many years after they were first activated. So again, the medulla, yes, that's where plasma cells are, but certainly not the only place where that occurs. So now we're going to have to look at this next slide, which is somewhat complex. And I don't want you to get confused here. I want you to pay attention first to the right side of this cartoon, and then we'll look at the left side. Now, on the right side, what we're going to be looking at is how B cells snag antigen. Now, up until this point, I've told you that B cells can snag soluble antigen without any help whatsoever, and that's true. Small antigens can be recognized by the antibody on the surface of the B cell, and then be endocytosed, etc. However, more often in the lymph node, what happens is antigen is brought as if it were on a ferry. And the ferry could be a dendritic cell or some other type of antigen-presenting cell. And in this case, don't get the wrong impression. The antigen is not endocytosed or presented with MHC class II. I'm not talking about how we activate T cells. In this situation, antigen binds to the surface of a dendritic cell and can do so especially if the antigen has activated complement through the innate immune response. Complement particles can cling to that antigen molecule. And dendritic cells actually have complement receptors, so the antigen and complement can actually stay on the surface of the dendritic cell or be endocytosed. So they can be both places. So we have the dendritic cell acting as a ferry to bring not processed antigen, although it can do that as well. It can do that as well. But cell surface associated antigen dragged into the lymph node. In addition, however, free antigen, if it's small enough especially, can also enter the lymph node. Now, I want you to read through phases 1, 2, 3, 4, and 5, and then I'm going to discuss them with you and try to make some sense out of it. I want you to read through it because I know you just had an exam last Thursday, and I bet you dollars to donuts, no expression if I may use. None of you have looked at these slides, and most of you haven't. So I want you to look at steps 1 through 5 on the right for processing by B cells, and then I will return to your conscious domains in about a minute. SCSM is subscapular sinus macrophages. Subscapular sinus macrophages right here. It's the subscapular region. Oh, right here, sorry. The capsule of the lymph node, right beneath the capsule of the lymph node. That's what that means. That blobby thing sticking out is the afferent lymphatic. This thing's afferent lymphatic. All right, that's enough time. Now, what happens here? The overarching principle here is that this antigen is handed off to various cells in the lymph node, eventually working its way to being recognized and snagged by antigen-specific B cells. That's the ultimate destination of these antigen particles. Now remember, don't mislead yourself. We're not talking about antigen processing. where we're considering class 1 or class 2 yet. It's just all surface associated, cell membrane surface associated. So what happens is the dendritic cell drags in antigen, or antigen could be coming in by itself if it's small enough. And there is some evidence that the dendritic cell surface antigen can then be handed off to macrophages, which are SCSMs, as labeled here. And what happens after that is ambiguous. Your book states that any B cell with a complement receptor then can snag onto this complement antigen complex, which makes sense because it's not antigen specific. And then that B cell, then they claim, and I'm not sure this is actually the case, hands off the complement and antigen to a very important cell, and this does happen. Ultimately, this is the cell the B cell's probably going to interact with the most. Follicular dendritic cells, FDCs, follicular dendritic cells, one way or the other. The antigen brought in by the afferent lymphatic, whether free or originally brought in piggyback on the surface of a dendritic cell, eventually whether or not these nonspecific B cells are involved is debatable. Nonetheless, eventually the antigen plus complement ends up on the surface of these very important cells called follicular dendritic cells. Now, follicular dendritic cells are not bone marrow derived, nor do they process antigen. They don't then endocytose this antigen presented with class 2, if you will, or if they were infected, present with class 1. These are not the traditional dendritic cells. It's unfortunate they have that name dendritic cell because they're not related to them whatsoever. They're not developmentally related. They're not any way really related. However, they look like dendritic cells, and they reside in the follicle. So don't make this mistake in your minds. These are called dendritic cells because they have these things sticking out like dendrites, just like our normal dendritic cell, but they're not related to it. And they're called follicular because that's where they live, in the B cell follicle, if you will. So they're called follicular dendritic cells. Their main purpose, if you will, is to display unprocessed antigen on their surface. Don't get mixed up. I'm not talking about processing again. It's unprocessed antigen on the surface of this dendritic cell, follicular dendritic cell. And the final nail in the coffin for not being related to traditional DCs, they're not even derived from bone marrow cells. Okay. Now, these follicular dendritic cells eventually will then attract antigen-specific B cells, which will then, once they bind specifically to the antigen, be able, through their antibody on their surface, not through the complement receptor. That's the difference between these B cells and these B cells. These B cells are recognizing complement plus antigen. This is recognizing antigen specifically through the antibody on its surface. Once that occurs, this antigen-specific B cell will then endocytose the antigen, process it, and present it with class 2. Get the difference? So these antigen-specific B cells will process the antigen if they bind specifically via the antibody molecule. All right, so that's what happens on the right side. Now I'm going to look at the left side, because we have to have T cells help the B cells produce antibodies. And now I'm going to look at the left side. Why this is a good diagram, because we're going to combine the role of both T cells and B cells in helping B cells produce antibodies, and follicular dendritic cells as well. So we'll look what happens here. Ultimately, there are 4 different cell types involved in antibodies being produced, 5 actually, if you include plasma cells from B cells. All right, so what happens on the left side here? We're getting T cells and APCs, dendritic cell in particular, congregating together. So in this case, we have our afferent lymphatic, and what happens in the periphery before we get into lymphatic circulation, we have antigen and dendritic cells interacting with each other. And through TLRs, then activation of phagocytic receptors, recognizing some PAMPs, etc., which we talked about the first week of class. Nonetheless, the antigen is endocytosed by the dendritic cell, processed, and presented with class 2. And potentially, if it's cross-presentation with class 1 as well. Correct? Then, because of the infected area becomes inflamed, we have increased blood flow to that particular area, increased leakiness of the blood vessels. That leads to more extracellular or interstitial fluid being produced. Remember that from the very first part of the course. That increase in interstitial fluid creates a pressure gradient, which pushes the antigen and the dendritic cell into the afferent lymphatic, and now we're draining into the draining lymph node, the one nearest that afferent lymphatic vessel. So think of all those steps we're putting together now in the course, how we're going to get all these different ingredients together to produce a B cell, which can lead to a plasma cell producing antibody. Right, so now we get into the lymph node. The antigen-presenting cell then works with the reticular network of the lymph node, especially in the paracortex, which directs the dendritic cell to the correct area, paracortex in this case. So it's called a fibroblastic reticular cell network, which we won't worry about really at all in this class. And then we have the T cells here as well. And if the T cell recognizes through its receptor, TCR, the correct class 2 and or class 1 molecule, depending upon whether it's CD4 or CD8 cell, it'll get signals from the dendritic cell to become activated, and one way or another, which we'll talk about next week, or next Thursday rather, and the T cell will then become activated. So this primary interaction between the T cell and the dendritic cell leads to primary activation of this T cell. And we're going to pretend our T cell today, our T cell du jour, is going to be the follicular helper T cell. Follicular helper T cells are CD4 positive, and their main function, if you will, is to help B cells give rise to plasma cells. So that's what we're going to be looking at. However, as an aside, and the same locus, we may have a dendritic cell activating a T helper 1 cell. In addition, this dendritic cell might also, by cross-presentation, be presenting antigen with class 1. And therefore, we get a CD8 cell also binding in this area. So it's in this region where we get a CD4 cell, not shown here, helping a CD8 cell become active to be a cytotoxic cell to destroy virally infected cells. So all this is going on in the paracortex. But today, our interest is going to be only pretend these are follicular helper T cells. Remember, the cast of characters for T helper cells is huge. So we're going to limit our discussion mostly today to follicular helper T cells. And anytime I use the word helper, immediately assume, because it's always correct, these are CD4 cells. These are CD4 cells, not CD8 cells. All right. All right, so now I've set the scene, almost set the scene for how T cells are going to help B cells produce antibody, and we're now going to develop that story. We already learned how antigen and antigen-presenting cells get into the lymph node through the draining lymph node, but how in the world do the lymphocytes get to the lymph node? Lymphocytes get to the lymph node through high endothelial venules. They get there through the blood circulation initially, not through afferent lymphatics initially, or the bulk of it. And the reason is T cells express a molecule we talked about already, CD62L, which is also called L-selectin. And L-selectin is a protein which likes to bind to sugar. And the sugar this one likes to bind to in particular, L-selectin, is sialic acid. S-I-A-L-I, not salicylic acid. No, it's not aspirin. It's sialic acid, S-I-A-L-I-C. And in particular, on HEVs, this is not HIV, this is HEV. Big difference. There's a sugar called glycan, G-L-Y-C-A-N, which is highly decorated with sialic acid. And again, naive T cells will bind to that through CD62L. That interaction will drag T cells and B cells out of the bloodstream into the lymph node. So again, we can drag if you think of magnetically, lymphocytes, both T cells and B cells, out of the bloodstream to the high endothelial venules into the lymph node, where, again, through chemokine receptors and chemokines, they work either the way to the paracortex or to the cortex. Cortex if it's a B cell, paracortex if it's a T cell, and also both can go in both directions too. All right, so we set up the picture, I think, for how all the cells and the antigen get to the lymph node. We have all the ingredients. We have the sugar, we have butter, we have flour, we have eggs, we have a pinch of salt, a little bit of nutmeg, apples, and now we're going to make apple pie. Yes, question.
So do the lymphocytes literally bind to the sugar, or is they just?
Yes,
how does it work?
they literally bind to the sugar.
Through what though? Is it
Through the ligand, because CD62L is like a magnet being attracted to an iron filing. It's the same type of thing, just does bind to it through non-covalent interactions. Okay. Not magnetic, but you can think of it that way if you want. All right, any other questions? Yeah.
Did you say L-selectin is on B cells and T cells?
Right, yeah. So we have it on both. So I always bias my discussions to T cells, because I'll be, like any parent, I try not to divulge who is my favorite child, but I really do like T cells more than B cells, so I tend to overemphasize T cells, but B cells also, same thing. Okay, other questions? I only have one kid, so it's not a problem for me. So, other questions? All right. Now remember, the mystery in all of this, how the cells know exactly where to go, that's again the upregulation and downregulation of receptors and chemokines being produced and dragging them to one place, then at another time taking them to a different destination within the lymph node. So that's the real intellectual exercise to do, and that's the real experimentation to do in a laboratory, that's causing this upregulation and downregulation, and that's still unknown. That's where to go, in case anyone wants to pursue this for a career. That's the future. Rather than artificial intelligence and computer science, this is the future for immunologists. Now this is just showing you a slapdash cartoon of how a B cell can imbibe, if you will, antigen on the surface of an APC. Now I want to emphasize one more time, do not draw the wrong conclusions from this slide. We're not talking about processed antigens. This is a different story than we've adhered to up till now. In this case, the antigen is superficial. It's intact on the surface of the APC, probably bound through its complement and a complement receptor on the APC. Yes, some of it is endocytosis imbibed, but a lot of it stays stuck on the cell surface. Again, it's not all or none in an immune response. Yes, some that gets endocytosed and expressed with class 2, but a lot get just stuck on the cell surface, which then can be recognized by a B cell, either through the complement receptor on the B cell I just described, or more hopeful through the antibody molecule on the surface for antigen specificity. Okay, get the sequence. And again, this is just clathrin-coated pits, and you probably have heard of before. If you've taken Bio 13. If you haven't, don't worry about it. Did Susan discuss clathrin-coated pits in Bio 13? Yes. But if you haven't heard about it, don't worry. It's not worth it. Now, here's our primary follicle in the paracortex. Here's the germinal center, and here's the plasma cell. There's 3 stages we're going to look at in detail today. Then we got the primary follicle. The primary follicle is where you first activate the B cell. That's why it's called the primary follicle. And that occurs in the paracortex in the predominantly T cell area. But you can see already it's not just T cells, because we have B cells in the paracortex. These T cells, and again the FH T cells, and by the way, very often on exams and whenever I type things out, I put capital T subscript, not subscript, just lowercase FH, because I'm too lazy to do the subscript for capital FH, so I often abbreviate my cells capital T, lowercase FH, just because I'm too lazy to make a subscript capital FH. So, because TFH would be something else entirely. All right, anyway, you can see how it's written, and that's correct, and very often I'm too lazy to do that. I'll admit, I'm lazy. So, what's missing from this story is we have to get the T cell first activated. So our first story is to get this T cell activated, and that occurs in the paracortex. And what happens in the paracortex is we have the T cell. And what T cell am I talking about in particular? CD8 cell? Am I talking about a CD4 cell? Yes, don't fall into that trap. But what type of CD4 cell am I talking about? Follicular helper T cell. And the follicular helper T cells first has to be activated by binding to the dendritic cell in the paracortex. But in this case, T cells hate unprocessed antigen. They can't recognize it. So for this story to be started, we have to have the follicular helper T cell recognize processed antigen on the dendritic cell. Remember I told you, dendritic cells enter the lymph node, and they have antigen on their surface. Some of that gets endocytosed and processed. A lot of it stays on the cell surface. So we have both ways we can decorate our dendritic cell with antigen, unprocessed and processed. Two ways we can present it. That's important to keep in your mind. All right, so this is a pre-activated TFH, and it binds now to the B cell. Now, how's it going to bind to the B cell? Someone tell me, how's this TFH going to bind to the B cell? How's that going to happen? Phenomenologically, how's that going to happen? I really told you how that happens. How, but you now tell me how that happens. How does that happen? How does a TFH phenomenologically, nice word, bind to the specific B cell? Well, I saw your hand first.
Well, once the antigen-specific B cell endocytoses and processes the antigen, then it can present it with the MHC class 2.
Right, stop, you're correct. Stop, the reason I'm stopping you is I want to put a little more detail on it. It's absolutely correct. By the way, we have a microphone here. Does this work? All right, let me do my talk show host routine here. So, where did the B cell first encounter antigen? Where did the B cell first encounter antigen? Probably in the cortex. Okay. So what was the cell that the B cell first encountered antigen with? Might be a follicular dendritic cell. Remember follicular dendritic cell? Now, the follicular dendritic cell, true or false, only presents processed peptides. False. I say that very loudly. Hello? Okay, false. What it does is it's decorated on the cell surface, and the B cell can take that up through its specific receptor, the antibody on its cell surface, imbibe, endocytose that antigen, process it, and then present with class 2. The B cell first is in the cortex. So here's our first journey that we have. The B cell, when it leaves the bloodstream, goes to the cortex preferential, not the paracortex. Get that? Once that happens, the B cell then wends its way into the paracortex. How does it do that? Alana, how does it do that? Allegra, how does it do that? Janine, how does it do that? The upregulation, I'm giving you a hint here, upregulation of a key.
Of chemokines.
Yeah, you got it right. Chemokine receptors and chemokines, right. So remember I told you it's going back and forth. It's not magic, and that's what the rich area of research is. looking at the upregulation, downregulation of chemokines and chemokine receptors. So the B cell going back and forth, back and forth, back and forth. So if you're disturbed by that, drop the course. That's the way it is, folks. The thing moves back and forth. B cells continues to move back and forth. It's not a static type of situation. It's very dynamic. All right, enough playing host here. All right, so now we're back in the paracortex. The B cell was taking up the antigen, processed it, presented with class what? Class MFU class, presented with which class? 2. Great. You're almost at the end of this course. I hope you do that. Class 2. And then we get a hookup between the FHT cell and the B cell. How? Look at that slide for 15 seconds. What is creating this specific hookup between the TFH and the B cell? Again, FH, remember, is follicular helper cell, which is distinct from the other CD4 cells I told you. This is really important because now we're going to get specificity, because this B cell can only get T cell help from a real small segment of the total follicular helper T cell population. That's real important to know. How's it going to do it? Alana, how's it going to do it? Since you started the story.
Is it the P-SMAC?
Well, yes. There's a P-SMAC and a C-SMAC, but right. Well, it's mostly the C-SMAC, but go ahead. As long as you brought that name up, it's mostly the C-SMAC, but what's happening?
So the TCR recognizes simultaneously.
Now look how fluent we all are in this stupid language in immunology. She said TCR as if she is a veteran, if you will, of immunological laboratory research. Yes, go ahead.
So it recognizes simultaneously the MHC class 2 and also the processed peptides.
Exactly. So is this going to be a specific or promiscuous interaction?
Specific.
Right. It's not going to be just like MHC can bind many molecules. Since the TCR is very specific, what it can recognize by specificity and restriction, it will only bind to a B cell, which has the right restriction, which we're not going to worry about because this is all done in one animal. I'm sure that will take place. And the right specificity. So the B cell also has to have the right processed antigen on its surface to get help from this particular follicular helper T cell. Okay. Now, what else is happening? Someone else. What else is happening? Besides getting the C-SMAC, that is the, remember, the immune synapse in the center, which gives us the specificity. We also have the P-SMAC, which is the periphery, which gives us other molecules which increase the avidity of the T cell to bind, in this case, to the B cell. So what are those molecules in the P-SMAC? Look at the slide. Look at the slide. You're not going to get the answer unless you look at the slide. What? CD40. We have not introduced to this molecule yet. There's CD40, okay? And the T cell produces a molecule called CD40 ligand. We're going to come back to this molecule probably pretty continuously until the end of the term. CD40 ligand is really important. It's upregulated on an activated T cell. Now, I just told you, you have to think of all these steps. I can't do them all simultaneously. You have to keep on reviewing them in your head. How did the T cell express CD40 ligand? Now, how did it first become activated? How did this T cell first become activated? Remember, you have to review all these steps. So, we have CD40 ligand, yes. Yes, the dendritic cell. Where did that take place anatomically? Sorry? Not the cortex. Paracortex. That's only in the paracortex. Now you have the paracortex where you get the T cell first activated. And what did that mean? I didn't tell you, but now you know. One of the hallmarks of an activated TFH is that it'll sprout this very important molecule, CD40 ligand, which has its own CD number, by the way, which I forget. But anyway, CD40 ligand is one still mostly used. And CD40 ligand, in turn, not too surprisingly, binds on the B cell via CD40 on the B cell. So this acts in the peripheral supramolecular activation complex, the SMAC. Okay, that's again the immune synapse. And we get, therefore, more tight binding between the FH and the B cell. And there's something else shown here. They put ICOS, ligand, and ICOS. I didn't tell you about ICOS. ICOS is similar to CD28. Actually, I did tell you this. But it's usually not expressed by naive cells. It's only expressed by cells which have already been activated. And again, where would we get ICOS presented on this FH cell? In the paracortex, once the FHT cell engages antigen process on the dendritic cells. Let's review all this, because I know there's a lot of steps here. And constantly you should think to yourself, how did this T cell get to the lymph node? How did the antigen get to the lymph node? And where did the first activation of these cells first take place? All right, where did the B cell snag antigen? Let's review that quickly, because it's important you don't lose sight of all these important steps. Where did the B cells first snag antigen? What part of the lymph node? The cortex or the paracortex? Cortex. Right. Where did the FHT cell first engage antigen? The cortex or the paracortex? The paracortex. And what cell in the cortex activated the B cell as far as antigen goes? Ultimately, what cell did we first learn about today? A new type of cell. What was the name of that cell? Follicular dendritic cells. Now what about in the paracortex? The T cells were activated, the follicular helper T cells were activated by primarily dendritic cells, and we already examined how they all got where they're supposed to. And now we're back in the paracortex. The B cell wends its way back to the paracortex after snagging antigen in the cortex. And now we get the B cell activated. What does that mean, we activate the B cell? Now we have to know, what in the heck does that mean? When I say we activate a B cell, the following two phenomena take place. When the B cell and T cell first engage each other, now again, where the first time the B cell and T cell are going to engage each other? Anatomically in the paracortex. What happens is, because of the P SMAC and the C SMAC, as long as we remind us of that immune synapse acronym, the B cell starts transcribing, translating, and presenting on the surface cytokine receptors. So this entire interaction you see in the primary cortex causes the B cells, not shown here, to start producing cytokine receptors. Not shown here. The receptors aren't shown. Simultaneously, or almost simultaneously, the T cell in concert starts pouring out cytokines which bathe the B cell that is connected to it. So in this primary focus, we're getting the B cell to express cytokine receptors. I told you this is a lot of complicated steps. The FHT, but my 7-year-old brain can still process all this. Yours is much more agile. And the FHT cell starts pouring out cytokines, which then fall on the cytokine receptors, and transcriptionally and translationally start changing the B cell's phenotype. That's what we're going to say in a moment. And what that's going to do is going to push the B cell into the stage of the cell cycle where we're going to have mitosis, we're going to have replication of DNA, etc. So we're going to have expansion occurring. And when that's happening, the B cells now move back to the cortex. Remember I told you it's confusing so much when we're going back and forth? The B cell was first in the cortex, moves to the paracortex, and then back to the cortex. And how does that somewhat magical set of steps takes place? Because of the upregulation and downregulation of what? Cytokine receptors and chemokines, really. Chemokine receptors and chemokines. Okay. So now we're back in the cortex. And we are now in a germinal center. Remember I told you a germinal center is a secondary follicle, center of a secondary follicle where proliferation of the B cells are occurring. Everyone with me now? Steps so far? Okay. So in the germinal center, we have two main regions. We have a dark zone and the light zone. Now, what would be your educated guess of the dark zone or the light zone? Where do you think you have more packing of B cells, dense packing of B cells? In the dark zone or the light zone? Where do you think you have denser packing of the B cells? Dark zone or the light zone? Microscopically. Dark zone. So what's going to happen now, this is you're probably going to blow your brains out with this. The B cells proliferate to a great extent in the dark zone, then eventually move to the light zone, and then move back to the dark zone, and then move back to the light zone. Back and forth, back and forth, back and forth. Now, why that is evolutionarily advantageous, we're going to learn today. But you should already figure out how that happens. How does it know how to move back and forth between the light zone and the dark zone? Because of upregulation and downregulation of chemokine receptors and chemokines being produced, okay? All right. Now, just to make things less ambiguous, if you look at it this way, or more complicated, if you look at it another way, B cells aren't called B cells anymore in the germinal center. They undergo a new name. They went to the courthouse, they put in an application, they wanted to change its name, when they were given at birth, and now they're given a new name. They're given centrocyte or centroblast. Centrocyte or centroblast. Now, not plasma cells yet, then eventually. So centrocyte or centroblast. Now, that suffix blast always indicates rapid mitosis. So unfortunately, leukemic cells, if you see blast cells in a leukemic patient, that's a bad sign, because that means you're getting rapid proliferation of leukemic cells, and you do not want to see that. But leave that pathology aside, the suffix blast means you're getting proliferation. It's blasting. It turns out in the dark zone, our cells are not called B cells, they're called centroblasts. They're going rapid proliferation. Once they move into the light zone, that blastic stage ends, temporarily at least. So we're given a new name. They're called a centrocyte. Centroblast and centrocyte. Not to confuse you, but this way we can be more specific of what cell I'm talking
So they don't move back and forth.
about. No, they do. They move back and forth. Yeah. It makes it even more crazy. So you have a centroblast move into the light zone, become a centrocyte, and then after a while you'll see why decides, I'll do my air quotes, decides to move back into the dark zone, and then now it's called the centroblast again. So it moves phenotypically from the centroblastic, where it's undergoing, MS, etc., for the cell cycle, and back to the light zone, the centrocyte, where it's more quiescent as far as cell cycle goes, G1.
How did the B cells end up in the cortex in the first place? Is it because of the
Chemokine and everything. Always that.
Oh.
Always that. Yep. Always. No exception. All right. So now in the germinal center, collectively, but not in both places, not in the dark zone and light zone collectively, but we'll learn that in a minute. We undergo somatic hypermutation.
Where does that happen?
In the germinal center. I'm not telling you where yet. That's to come in a minute. And then affinity maturation, which means you select the B cells which have the highest affinity for the antigen and get rid of the others. And also isotypic switching. All three things I alluded to or told you explicitly what happens to a B cell during its life is now going to take place in the germinal center, either in the dark zone and or in the light zone. So our next task is to differentiate where these different processes are occurring and what's bringing them about, and what are the consequences of them. All right? So what I want to get in your minds, though, anatomically, where everything's happening. So again, I know I may be boring you, but I'll do that anyway. I'll list that because I want to make sure you have all this. Otherwise, the rest of this lecture won't make any sense. Where do B cells begin their activation process? In the lymph node, in the cortex or paracortex? Cortex, right. And who do they first encounter there? What type of cell? FDC. Make sure you say the right cell. Don't just say APC. You'd be incorrect in this case. You have to say FDC. Very unique type of cell. It's not really considered an APC because it doesn't process the antigen. So now you have the FDC, and the FDC. How did the FDC get the antigen on its surface? Were there magnetic filings on the surface and the antigen had iron particles on it? How did it get there? Because the antigen might have activated complement innately, remember. So we still don't have the adaptive immune system upregulated. All right, and then the B cell snags the antigen specifically, one way or the other, then processes it and presents it with class 2, moves into the paracortex, encounters the FH. But where did the FH cell, what did it first have to do? And where did it happen? Where did the FH cell first have to do? It had to become activated. What activated it? Dendritic cell. Where did that occur? Paracortex, right? And what do I mean activated? Tell me anatomically, phenomenologically, what happened to the T cell once it was activated? What changed about it? It expressed what? CD40 ligand, right. Now the CD40 ligand via the SMAC can line up with the correct B cell, antigen-specific B cell, and you know now that the B cell will be antigen-specific because it only recognize a T cell which has processed the antigen that it can recognize, right? Because remember, the B cell processed the antigen of class 2, so I think that the TCR hooked up to the B cell specific, correct? Then, what does the B cell sprout? CD40 and what else? Cytokine receptors, and what does the T cell start doing? Throwing out what? Cytokines, which now bathe the B cell in cytokines, and now cause the B cell to start to do what? Proliferate. And where does it move after this primary focus takes place? Where does it move? Into the germinal center in the cortex. Okay, so back and forth. It goes back and forth. Don't be confused by that. It does go back and forth. All right. Eventually, by the way, after all this blah, 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 blah occurs, you end up with the plasma cell. And where's the plasma cell end up? The plasma cell ends up in the medulla, and eventually many of them in the bone marrow, maybe perhaps for decades, dumping out antibody, protecting you from a second infection. And I typed that out there for you. Okay, everybody with me? I know it's complicated stuff. Everybody with me? I could have typed this all out for you like I do in Bio 14, but heck, I'm not going to do it in a 3-digit course. So there you go. Leave Bio 14 behind you forever and ever. I know I have. All right, anyway. All right, so oh crap, I did tell you. All right, anyway, I did type it out. I didn't realize it did. All right, anyway, yes, Alana.
When the complement activates it, how does the FDC get it before the complement lyses it?
Because these are complement fragments. Remember the complement fragments like C3 complement? Yeah, like C3D.
Yeah.
That's what we're talking about.
C5A.
Right, no, C3D. We're talking about C3D. I told you that once in passing, so it's not. We're not going to We're not going to go to the MAC in this case. All right. So here's what it looks like: the dark zone, the light zone. There's a mantle zone too, by the way. You can get a disease actually, the mantle zone. I'm not going to tell you about that, which is quite lethal. All right. And anyway, dark zone, blah 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 blah. Okay. The dark zone contains centroblast, which I told you. Okay. All right. FDCs are found mostly in the light zone. All right. I told you that as well. I think. Did I tell you the dark zone a somatic hypermutation takes place? That's where you get in the high rate of mutation. All right. And then the centroblasts lose class 2 and no longer present antigens, so they're lost T cell help temporarily. And then they move to the light zone, become centrocytes, and now they re-express class 2, and then they compete for antigen that is concentrated in the light zone. Now what happens here in the light zone? All right, let's think about this. We have somatic hypermutation in the dark zone. Again, what enzyme drives somatic hypermutation? You had this on the first exam. What enzyme drives somatic hypermutation? Begins with an A. Activation-induced deaminase, all right, in the B cell. That gets activated, so now we get somatic hypermutation, and you only mutate probably on the CBR1, 2, and 3, because you have the right nucleotides associated with those sites to lead into a high mutation rate. We also did that for the first exam. And now we move to the light zone. We express class 2. And now honestly, I want you to talk with your neighbors and figure this out. I'm going to give you a question or a puzzle here, and I want you to figure it out. The central blast moves back to the light zone. We express class 2. And remember, you came from the dark zone where you had high mutation rate. And in the light zone, remember, you have dendritic cells, a rich number of dendritic cells, which have antigen on their surface. You also have lots of FHT cells. You figure out how all those ingredients can lead to the selection of B cells which have higher and higher affinity for the antigen. Discuss it. See if you can figure out. All 3 cells are going to be involved. Our B cell, our follicular helper T cell, and our follicular dendritic cell are all going to be involved in selecting for B cells which just by accident mutated to have a higher affinity for antigen. Remember, the mutations took place in the dark zone. And in the light zone, we're going to get affinity maturation. We're going to now select for the B cells which just by accident mutated to have a higher affinity. Remember, because of the degeneracy of the genetic code, when you mutate, you can have the same affinity, you can have lower affinity, or you can have higher affinity. The job of the light zone is to select those B cells which only have higher affinity. See if you can figure it out. Go ahead. We'll hear some discussion here. I'll give you a hint. I'll give you a hint because I'm not sure you can figure this out. So I'm not sure I would have. I forget, maybe. The follicular dendritic cells do their role first, followed by follicular helper T cells. That's the hint. Follicular dendritic cells do their job first for affinity maturation, and follicular helper T cells do it second. I'll tell you that. Go ahead, keep on talking.
I'm saying that with more and more proliferation of people
It's for now.
This is all the cortex, right?
Brain germinal.
So germinal cells still in the cortex.
Subdivide. And a map.
Well, then where does the B cell take place, right?
Remember starting the cortex and moving to the paracortex?
Yeah, but like cortex or like where they start
Oh, they just start in, primary follicle. Not until they get actually first paracortex back into the cortex. They start getting secondary follicle.
or they form the follicle.
And then the primary focus. All right, I don't hear any more discussion. All right, class. So, did anyone figure out what happens? Pick out the B cell with the highest affinity. The order of sequence. Remember I told you which cell do you start with? I gave you that hint. Which cell do you start with? FDC. Anyone have an answer? You have your hand up?
So B cell mutate function of that
Right.
will come up to try and grab antigen from the FDC,
That's right.
and the ones that do it the best will be able to process them, and this is for the helper T cell.
Perfect answer. Boy, that is absolutely succinct and accurate. By the way, in your answers on exam, those people who write the most usually get the fewest points because you don't know what you're talking about. You just go on and on and on. And if I were a real creep, I would deduct for extraneous information in some of those answers, but I don't. Especially ticked me off this year in Bio 115. People just wrote, don't throw the kitchen sink at me. Be very specific what I ask. Answer very specifically, all right? Because I have to grade 171 of these things, and extraneous information means you don't know anything. You're just throwing everything at it and hoping something sticks, all right? Anyway, exactly right. I'm giving you 10 out of 10. Let's figure this out. I'll repeat it anyway. So now we have the B cell back in the cortex that's been pre-activated from the paracortex. What's that called when it's pre-activated in the paracortex? The primary focus. The primary focus, right, in the paracortex. And remember, it got some help from cytokines produced by the follicular helper T cells. Now these primary focus B cells migrate into the cortex, undergo a somatic hypermutation in the dark zone. They create a dark zone because it's proliferating so greatly, and they're called centroblasts. And then they move into another area high on top of it, of the germinal center, called the light zone, because there's fewer cells. And now here, the B cells, which just mutated by accident to have a higher affinity, can outcompete B cells which have lower affinity antibody for the limited amount of antigen on the follicular dendritic cell. Remember, the follicular dendritic cells are decorated with the antigen, and the B cell can recognize just unprocessed antigen. But if it's now playing musical chairs, the B cell which mutated to have a higher affinity antibody on the surface will outcompete the B cell next to it, which has a lower affinity antibody, or the same as it did before, and therefore will snag up more of the antigen and therefore process more of it with, and then present with class 2. In order to keep the B cell going, it needs lots of help in the B cell follicle to continue. It needs continuous T cell help, not just in the primary focus. It needs continuous T cell help. And then in the germinal center in the light zone, we also have TFHs. And those B cells which outcompeted their fellow B cells, or centrocytes, I should say, for the limited antigen on the dendritic cell, now we receive additional help from the additional helper T cell, more cytokines out. Okay? And therefore will survive. The other B cells will die from lack of attention, undergo apoptosis, won't upregulate BCL2. Those which do bind to follicular helper T cells will express BCL2, preventing caspase cascade, which leads to apoptosis. And then we just drive evolutionary. This is a Darwinian selection. It's Darwinian and evolution in its truest form. B cells which have higher and higher affinity. And that means over time, your antibody to a particular antigen will have a higher affinity, and you'll be able to dispatch that antigen with alacrity on more speed the second, third, and fourth time you encounter that microbe, because your antibody keeps on mutating to have a higher and higher affinity. And again, it's all a Darwinian selection. So if you're ever educating people about the truth of evolution, all you have to do is look at the lymph node. We're not creating new species, I'll admit, but we are selecting for B cells which have higher and higher affinity because of local environmental circumstances. Absolutely the case. So it's truly Darwinian. And then what you have then is you now have a B cell which has high affinity for the antibody, for the antigen, and this is not a one and done process. It's continuous. It goes on and on. Okay? So this goes on through your entire life. So I already introduced you to the concept of broadly neutralizing antibodies. Remember I talked about them for HIV. Some people through this process of affinity maturation, somatic hypermutation, just randomly, accidentally by a mutation, ended up producing an antibody which could bind broadly with high affinity to very sensitive areas on HIV, so it can no longer bind to CD4 cells. It's called broadly neutralizing antibodies, and that happens not because in the genome they had the right VDs and Js and VJs, but it occurred by somatic hypermutation, and they just lucked out. Their somatic hypermutation mutated them in that direction. So we'll talk more about broadly neutralizing antibodies when we do HIV. And then, by the way, as I said, once and done is not what happens. Because once this process of selection occurs, the centrocyte now goes back to the dark zone, becomes a centroblast again, undergoes another round of somatic hypermutation, and then goes back to the light zone, et cetera, and so forth. Okay? And you keep on selecting in a Darwinian way for B cells, the centrocytes that have higher and higher affinity for the antigen. It's an ongoing process. Okay? It's a beautiful system. All right, blah, blah, blah. I guess I wrote that out too. That's too bad. There it is. All right, this just summarizes, I didn't mean to write it out for you or type it out for you. I apologize for doing that because I wanted you to think, not me. And so you can expunge those slides from your PowerPoint set if you want. Rats, I can't believe I did that. That was unintentional. I forgot I did it. All right, at any rate, here's what's going on in a cartoon of what the heck these things do, blah, blah, blah. Okay. Now, how do you get a B cell turned into a memory B cell versus either short-lived or potentially long-lived plasma cell? It depends upon what transcription factors are present, and we're not going to discuss them. There's two different ones. There's BLIMP, which is this one. I like BLIMP. I like BLIMP because I like its acronym. I think it's funny. And the other one, BSAP, I gave you that acronym as well. BSAP will give you a more memory B cells, and the BLIMP will give you the plasma cell. And that's it, that's what I'm going to say. All right. Now, how do cytokines lead to isotypic switching? This is one of our last topics to talk about in B cell activation. Isotypic switching occurs mostly, if not entirely, in the light zone. It can only occur under the influence of follicular helper T cells. So we talked a lot today. So T cells again. Starring T cells again. Follicular helper T cells. And the follicular helper T cells produce different cytokines. And if you remember the process of isotypic switching, you had to have single-stranded DNA. Do you remember that? And then we got a process called sterile transcription, which means that no translation took place, but you have transcriptional activity occurring. And when you have transcriptional activity occurring, it means you have single-stranded DNA. At that point, the AID and the subsequent enzymes can then interact or act sequentially, and then you end up cutting and splicing out the exons which code for the upstream isotype, and then go to the ones which are downstream. And different cytokines favor different switch regions. The one which is most known is IL-4. Interleukin 4 will give you the switch to isotype IgE predominantly, maybe a little bit of GD1, but mostly IgE. The reason people are interested in that, of course, and I think I already mentioned this, IgE is the isotype associated with allergies, peanut allergies, all sorts of different types of allergies that you may have. IgE is the one. So interleukin-4 is the master cytokine switching in the light zone transcription away from the other isotypes upstream to this one, IgE, which is downstream, and cutting out all the other exons which code for the other isotypes in between. And then, again, I'm not going to have you memorize the different cytokines that are involved. The only one really which is interest to me clinically is the one for IL-4. The other ones are interesting too, but the IL-4 has the most clinical interest. So that's, I guess I can't give you more of a hint than that. Yes?
So is this like a random process in which cytokine?
I would. The question is a good question, and I'm going to phrase it slightly different. The question is whether this is a random process. We humans only say it's random because we don't understand the exact steps that are involved. I'll be candid. So at this point it seems random, but I bet you it isn't. All right, you know what I'm saying? There must be some environmental cues which are telling you predominantly which cytokine profile you're going to have in preference to another one to give you the isotype. I can tell you that if you have lymphatic tissue in the intestinal tract, undoubtedly the cytokine profile that you're going to favor is one that's going to give you IgA. You know that lymphatically. So for some reason in the mucosal tissue or the lymphatic tissue associated with the GI tract, you favor the isotype IgA, almost universally. Same thing in your respiratory tract, the same thing in your urogenital system. You're going to favor the IgA isotype. So anatomical location favors a certain type of profile of cytokines, and preferentially, again, in your in the parts of your body exposed to the outside world, your intestinal, respiratory, and urogenital, IgA is the favorite one to go. Okay. And in breast milk too, IgA is the favorite isotype that's produced in breast tissue. It's IgA. By the way, as long as I brought up IgA, IgA is important for the newborn in humans, which are different than mice. Need I say that again? In humans, the reason breast milk is really important to a newborn is because of the IgA for other reasons, but IgA in the milk coat the intestinal lining and prevent proliferation of bacteria in the GI tract. However, other mammals, breast milk also has IgG. And the IgG in other mammals is endocytosed in the intestinal tract and ends up in the bloodstream of the newborn. That does not happen in humans. So in humans, IgG would be, there is IgG in human breast milk, but most of it's broken down in the intestinal tract. In horses, IgG is in mare's milk, but that's endocytosed by the enterocytes, taken into the bloodstream of the colt or the foal, and ends up in the bloodstream of the newborn horse. That doesn't happen for whatever reason in humans. The most important aspect of the antibodies in human breast milk is the fact it prevents overproliferation of intestinal bacteria, which is no small thing. That's real important because you don't want the newborn to have, obviously, intestinal overgrowth of bacteria, since it can end up in life-threatening diarrhea. So that's something to think about. So in any case, so that answers your question, I hope. Thank you. All right. All right. Now, so far in our journey through the B-cell world, we talked about T-dependent antigens. Most antigens we described in this course are T-dependent. That is, in order for the B-cell to produce antibody, it needs T-cell help. But wouldn't you know it, nature makes it more complicated. Yes, most antigens are going to be T-dependent, but some are going to be T-independent, TI. And because that's the case, vaccines are going to be tailored to this phenomenon. I'll show you how in the next slide. But what is a T-independent antigen? T-independent antigen might be two different types. One is LPS. Okay, LPS combined to TLRs, certainly TLR4, and in enough enough LPS can cause the B cell to produce antibody. In this case, by the way, this is a polyclonal activator of B cells. LPS is a polyclonal activator of B cells. There's enough LPS around. You will activate non-specifically wide numbers of B cells with different specificity. So if you have septicemia, where you're getting uncontrolled proliferation, especially of gram-negative bacteria in the blood, you will activate many B cells non-specifically to start pouring out antibody. We also get a lot of other undesirable outcomes which lead to death. So in this case, this is called T-independent via LPS. And again, it's a polyclonal activator of B cells. The other type of T-independent antigen is one which has repeated epitopes. The epitope repeated, polyvalency of the epitope repeated many, many times. Good examples are pneumococcal bacteria. Pneumococcal bacteria have sugar on their surfaces, which is repeated many times. That can lead to cross-linking of a B cell, which has an antibody which binds to that sugar, and that will specifically activate that B cell to produce antibody. That's called T-independent antibody production. So completely independent of T cell health. Now, T-independent antibody situation does not have affinity maturation, does not have really isotypic switching, so stays as IgM predominantly. Doesn't again have affinity maturation. It's a weird type of situation. And it turns out children are pretty poor, young children, very young children are very poor at T-independent antibody production, which means very young children can be susceptible to bacterial infections because they don't have a way of activating B cells independent of T cell help, because they have a poor reaction to T-independent antigens. Consequently, humans devised a vaccine, and there's a bacterium called, this is not a virus, this is a bacterium, Haemophilus influenzae. I know it sounds like it's a virus because it has influenza in there, but this is a bacterium. And I want you to read this. I want you to figure out how the people figured out how to favor the production of antibodies in children who don't have good production of T-independent antibodies. This also works in older adults who also lag, whatever reason, an ability to respond to T-independent antigens. Go ahead and read this. There's a vaccine given to older people, pneumonia vaccine, actually which uses the same technique. In the case of adults, it's not, this particular bacterium is pneumococcus, but similar idea. So it has a lot of sugar on the surface as well. So go ahead and read this slide. By the way, these are called conjugated vaccines. If you ever see an ad on TV or an ad in a magazine, you'll see sometimes a conjugated vaccine. This is what it means. All right, what did the vaccinologists do here? How did they get the immune system to respond to Haemophilus influenzae in children, very young children? What did they do? Go ahead.
They checked the epitope that they wanted to inoculate, so carrier protein.
Right, okay, remember we talked about hapten carriers at the very beginning of B cell activation, so go ahead, right? So which would be the hapten in this case, according to our terminology? What would be the hapten? Right, be the polysaccharide. And what would be the carrier? Right, the toxoid protein. Remember, toxoid is not toxin. Toxoid, by the way, is a modified toxin which is not harmful, but similar. Okay.
So that gives it something to go ahead with the peptides.
Right, so what happens is the B cell specifically recognizes the hapten, or in case of pneumococcus sugar, or Haemophilus influenza sugar, takes up the bacteria, processes the protein for its class 2. Remember, class 2 does not present polysaccharides. And now the T cell, now you're fooling this system. Instead of being T independent, now you're making it T dependent. And now the T cell comes along, gives the B cell help via the carrier, ovalbumin, if you will, or toxoid, tetanus toxoid, or whatever the protein carrier is. And therefore really revs up this B cell to start producing plasma cells, which are very, very powerful in producing antibodies against the sugars on this particular bacterial surface. I said in elder adults, they do this for pneumococcus. It's a conjugated vaccine. It works quite well. It works well. And however, the problem is, will you have memory in this case? Perhaps not. Will you have isotypic switching? Perhaps not. Will you have affinity maturation? Perhaps not. But even with this vaccine, though, shows you how immunologists can be fooled. Children do have isotypic switching. How? I don't know. They have long-lived titers of the antibody, which means maybe there really is some memory involved here, or a heck of a long-lived plasma cells. So I really don't know. But in any case, there you go. So T-independent antigens are really interesting to vaccinologists, again, in both very, very young children and in elder adults, where this innate process doesn't work too well. This just compares primary and secondary antibody responses. Everything you should know at this point is just a good review. All right, see you on Thursday, and no promises about handing back your exams at that time.